So, students, today I will discuss with you about the in vivo gene therapy and the other forms of gene therapy that is given in your first semester syllabus in your GE paper, that is in your biotechnology and human welfare. So, let's get started now. So, about the in vivo gene therapy. In vivo gene therapy is the direct delivery of your therapeutic gene into the target cells of a particular tissue of a patient constitutes in vivo gene therapy. So in vivo gene therapy we are delivering the therapeutic gene into the target cells of a particular tissue. Now many tissues are potential candidates for this approach, where we are entering our therapeutic gene into the target cells of a particular tissue within the patient's body. And many tissues are involved for this in vivo gene therapy approach, like your liver cell, muscle, liver tissue, muscle, skin, spleen, lungs, brain, and blood cells can be employed for in vivo gene therapy. That means, suppose we have some disorder, we directly take out the cells from that particular tissue. Okay, not like that was ex vivo, sorry, not like that. We directly inject, we first construct a gene, our therapeutic gene, with other gene segments for proper expression of that gene, and we deliver that gene into the target cell of that particular tissue within the patient's body. So here we are not using multiple steps as we have done in ex vivo gene therapy, where in ex vivo gene therapy what we have done, first we isolate the tissue, we isolate the cells from the tissue, we culture the cells in the laboratory, then we transfer the gene, the desirable gene into that cells, then we check for whether the gene is getting expressed in that cells or not, and then we again reintroduce that cells, that transformed cells, that means the cell having our desirable gene properly expressed, into the target tissues of the patient. But here what we are doing, we are directly constructing a vector having our gene of interest and other gene segments for proper expression of that gene, and directly they are delivered into the target cells. Okay. Now, this type of gene therapy in vivo gene therapy can be carried out by viral or by non-viral approach. That means you can use virus as a vector, so that directly it can transfer our gene of interest into the target cells of the patient, right? Or non-viral approaches as well, like liposomes. Liposomes are artificially created lipid vesicles where we entrap our gene of interest within that lipid vesicles. Then that liposomes are allowed the process of endocytosis to engulf the liposomes by the cells, so that our therapeutic gene can able to enter into that target cell. Okay. Now, the success of in vivo gene therapy is mostly depend on the following parameters. So some parameters are there based on which your in vivo gene therapy depends actually. What are those parameters? First is saying that the efficiency or uptake of the remedial therapeutic gene by the target cells. We are transferring our we are actually delivering our therapeutic gene into the target cells. Now, what is the efficiency of the target cells that they will uptake that our therapeutic gene and properly integrate that gene into their genome of the target cell and gets expressed there? So depends on the efficiency of the target cells, where we are allowing our therapeutic gene to enter. That is one first problem, not problem, that is the first feature which has to be efficient, more efficient. If it is less efficient, then this type of approach will be some problematic. Second is intracellular degradation of the gene and its uptake by nucleus. So the next parameter is whether the therapeutic gene is getting degraded inside the cell or not after the cell have uh, properly uptake that therapeutic gene. Now the main problem is whether the gene is getting to be uptaken by the nucleus of that cell, properly expressed within the cell or not, or it is going to be degraded by the cell's enzyme, by the target cell's enzymes. So that is the second parameter of this whole in vivo gene therapy depends. The third is expression or capability of the gene, that how much the gene is gets to be expressed within the target cell. That's also another factor. It plays an important role. So three factors are there. First is how much the cell is uptaking our therapeutic gene when we are transferring into the target cell. Second is the cell is the gene is going to be inserted into the target cell. Okay, now whether the gene is going to be inserted properly, integrate properly into the nucleus or not, or is it going to be degraded by the cells own enzymes. Third factor is expression of the target gene. The therapeutic gene that we are going to be expressed, it's going to be expressed or not within the cell nucleus. Okay, so these are the three parameters based on which your in vivo gene therapy depends. Now in vivo gene therapy with special reference to gene delivery system, viral and non-viral, with suitable examples is being described. So first we will describe about how with the help of virus we can deliver our therapeutic gene, um, in this case of in vivo gene therapy. In ex vivo gene therapy we have used virus as a vector here also, but here also we will use viral systems for in vivo gene therapy. Let us see. So gene delivery by viruses. Many viral vector systems have been developed for gene delivery. This includes retrovirus. We have also used retrovirus system in ex vivo gene therapy as well. Adenovirus, adeno associated virus, and herpes simplex virus, which is known as HSV. So these virus can be used for in vivo gene therapy. Uh, like retrovirus we can use, adenovirus we can use, adeno associated virus, herpes simplex virus can be employed for in vivo gene therapy for transfer of our desirable gene of our therapeutic gene into the target cells or tissues. So first approach is by using retrovirus. So what it is saying here, replication defective retrovirus vector that are harmless are being used. So as I have mentioned with you during the time of ex vivo gene therapy, we can use retrovirus vector. We can use retrovirus virus as a vector, but it should be harmless. That means its pathogenic part has to be removed out. A plasmid in association with retrovirus, a therapeutic gene, and a promoter is referred to as plasmovirus. So we are converting the retrovirus into a plasmovirus. How? By using a plasmid in association with a retrovirus. Then we use a therapeutic gene and a promoter. Together, all of them together we call them as plasmovirus. So plasmovirus consists of a plasmid associated with a retrovirus. It will also contain a therapeutic gene within that plasmid and a promoter for its proper expression of the therapeutic gene during transcription. Together they are calling as termed as plasmovirus. The plasmovirus is capable of carrying a DNA therapeutic gene of size less than 3.4 kb. So that's as virus are very small in size, So the capability of carrying the target therapeutic gene is only out of up to 3.4 kilobase pair. Not more than that we can insert our therapeutic gene, okay, because of its small in size. Now those retrovirus which have defective in their replication machinery can be produced from the plasmovirus. So replication defective virus particles can be produced from the plasmovirus. As such, for delivery of genes by retroviral vectors, the target cells must be in dividing stage. It is saying that if you are using retrovirus as a uh, if we use retrovirus as a system for transferring our therapeutic gene through in vivo gene therapy, then the target cells where we are going to be inserting through the retrovirus, the target cell must be at their dividing stage. Okay, it is saying. But nowadays, in recent years, viral vector has been engineered to infect the non-dividing cells as well. Okay, so this is how your with the help of retrovirus gene delivery can be done by making them into plasmovirus. Plasmovirus is what? Plasmovirus is a plasmid associated with a retrovirus. Along with that, we have our therapeutic gene, and for its expression, we also use a promoter. Together, they are termed as plasmavirus, and they are delivered during the time when the target cell is at dividing stage. However, by some modifications, we can also insert our desirable therapeutic gene with the help of plasmavirus by to the target cells in non-dividing stage as well. The next type of viral vector system is your adenoviral vector system. Adenovirus with the DNA genome are considered to be a good vector for gene delivery because they can infect most of the non-dividing human cells. So those cells which are non-dividing stage, they can be um, what can I say? They can be infected by this adenovirus system. Okay, and that's the main reason that why adenovirus is used as a good vector for gene therapy, because they can infect most of the non-dividing human cells. A common cold and adenovirus is frequently used as vector. Nowadays, common cold adenovirus is used as a vector. Now it is saying that how they work. As the target cells are infected with recombinant adenovirus, the therapeutic gene enters to the nucleus, and there it expresses within the genome of the target cell. So when the target cells are infected with recombinant adenovirus, it is observed that the therapeutic gene gets properly integrated within the nucleus, and there it is expressing itself by recombination process as well. Obviously, due to with the help of recombination, there are various ways through which the DNA can be inserted, either by homologous recombination or by illegitimate recombination, which is totally different from homologous recombination. However, this DNA does not integrate into the host genome. Consequently, adenoviral based gene therapy require periodic administration on recombinant virus. But here it is saying, as I have not said, this DNA is not integrating into the host genome. That's strange. Well, for expression of the DNA, the DNA must integrate within the host genome. But here, in adenoviral vector system, the DNA is not integrating into the host genome. Thus, in this adenoviral gene-based gene therapy, it requires periodic administration of your recombinant virus. So thus adenovirus can be used as a good vector because they can infect most of the non-dividing cells in human. A common cold adenovirus is frequently used as a vector. Now, when the target cells are infected with recombinant adenovirus, the target DNA, the therapeutic gene, gets integrated within the nucleus and there it expresses itself. Okay, so this much you can write for avoiding the confusion that is given in the next line. Okay, now the ability or the efficiency of the gene delivery by adenovirus can be enhanced by developing a virus that specifically infect the target cells. Now the main thing is that um, if I want this adenovirus to be targeted among the other target cells as well, we need some helper virus as well. So the ability of a gene delivery by adenovirus can be enhanced by developing such a virus that specifically infect the target cells. Okay, so we can use this adenovirus so that large number of target cells can be infected by them and our allows our gene of interest, that is our therapeutic gene here, to be properly integrated within the target cells. And this can be possible by using an associated helper virus, as I have mentioned before, or with the help of a cell receptor protein. Okay, that is encoded by the DNA. Okay, so a DNA which is encoding a cell receptor protein can be employed for enhancing this process, because without interaction it cannot enter to the target cells. Okay, so this is about the adenoviral vector system, and I have said about the retroviral vector system. Now I will discuss about the adeno associated virus vector system. Now, adeno associated virus vector system is actually a human virus which can integrate into the chromosome number 19. Again, I repeat, it is a type of human virus. This adeno associated virus is a type of human virus which can integrate into the chromosome number 19. It is a single stranded, that means this adeno associated virus AAV, it is a single stranded non pathogenic small DNA virus of up to 4.7 kilobase pairs. As the adeno associated virus enters the host cell, DNA becomes double stranded, get integrated into the chromosome and expresses itself. So it is also can be used along with your adenovirus system vector system. We can also use adeno associated virus vector system, where this adeno associated virus, it is a non pathogenic virus. It is a single stranded non pathogenic virus whose size is not more than 4.7 kb. Okay, size refers to the DNA size. Okay, the size, the genome that is found within this adeno associated virus is of 4.7 kb only. And if we can use this adeno associated virus as a vector, then it will be very good, because it is non pathogenic. It has many targets, many target tissues. It has many target cells where it can able to infect. And as the adeno associated virus enters the host cell, DNA becomes double stranded. The genome of the adenovirus becomes double stranded, gets integrated into chromosome and expressed. So if we can somehow we can remove some unessential part, the non-essential part of adeno associated virus genome by our therapeutic gene, and properly integrate that adeno associated virus vector into the target cell, there it will insert its genome, okay, which also contain our therapeutic gene. It will become double stranded, gets integrated into chromosome and it expresses. Then it is saying that adeno associated virus can serve as a good vector for delivery of the therapeutic gene. Just now as I have said that we have to remove the non-essential part of the adeno associated virus gene, DNA. There we have to in that place we have to insert our therapeutic gene, and then we have to ligate it. Just we have seen in genetic engineering, we have seen by using restriction enzyme to cut, you properly integrate our therapeutic gene in that site, ligate them with suitable enzyme like ligase, right? And then we allow this adeno associated virus as a vector into the target gene. There, this virus will enter the host cell. The DNA within the adeno associated virus gets integrated within the host cell. It becomes double stranded, gets integrated into the chromosome and expressed. So thus adeno associated virus can be used as a good vector for delivering of the therapeutic gene. Recombinant virus are created by using two plasmid and adenovirus with the help of helper virus by a special technique. So now a special technique is being carried out by using recombinant. So we are using two plasmids, okay, and an adenovirus, that is a helper virus. By a simple special technique, we are using two plasmids and adenovirus together. Okay. Nowadays, some attempts have were being made to use therapeutic genes for the treatment of human disease like hemophilia, which is used for the production of blood clotting factor 9, and cystic fibrosis for synthesis of cystic fibrosis transmembrane regulator protein by employing adeno associated virus. So again I repeat, so with the help of adeno associated virus, uh, for curing two diseases has been done, have been a trial, and it has been very successful, like for cystic fibrosis, where a gene responsible for CFTR protein, cystic fibrosis transmembrane regulator protein, due to defectiveness of this CFTR gene, we can go for this adeno associated virus vector system curing that disease. And hemophilia occurs due to non-function of a clotting factor protein. Here blood clotting factor 9 is responsible for causing hemophilia. Due to the malfunctioning or due to non-functioning of clotting factor 9, it leads to hemophilia. And for this we can go for adeno associated virus vector system for curing this type of genetic disease. Okay. And in a special technique, as it has been mentioned, by using two plasmids and an adeno associated virus can be employed for this approach. Now let us see about the examples given for the in vivo gene therapy by using virus for gene delivery. Okay. Now let us see about the gene therapy, uh, the in vivo gene therapy of cystic fibrosis. Okay. Now let us see that what is cystic fibrosis actually and why this type of disorder occur. Cystic fibrosis is one of the most common and fatal genetic disease which occur among human beings. The ratio is 1 is to 2500. That means out of 2500, one person can have this type of genetic disease. Now it is characterized by accumulation of a sticky dehydrated mucus in the respiratory tract and lungs. So if we want to see that the patient who is suffering from cystic fibrosis, they have a sticky dehydrated mucus within their lungs. Well, mucus is actually in Hindi it's a balgam. Okay, বাংলা সর্দি বলে. Okay, now this mucus gets accumulated within the respiratory tract and lungs. Normally that mucus is a watery type of substance, but those patients who are suffering from cystic fibrosis, they have dehydrated mucus that gets accumulated within that respiratory tract and lungs, and it's actually a sticky type of thing. Now, patients of cystic fibrosis are highly susceptible to bacterial infections in their lungs, and most of them die before they reach to the age of 30. Now, those patients who is suffering from cystic fibrosis due to the presence of such a huge amount of mucus is always produced in within our body in order to clear away the dirt particles. Okay, but if that mucus gets dehydrated, it becomes sticky, then what happens? It creates a good environment for the other bacterial infections to occur within that site, within the lungs, and thus most of the people die who is suffering from cystic fibrosis. They die before they reach to the age of 30. Cystic fibrosis can be traced in European folklore in the following statements used to be said: woe to that child who which when kissed on forehead taste salty, he is witched and soon must die. So it is a folklore which is oftenly said by the European people. It means to say that, according to this, that woe to that child which, when kissed onto the forehead, tastes salty, he is bewitched and soon must die. So it is their folk song where they used to say about this cystic fibrosis. They have mentioned it. But the main biochemical basis we will discuss, that why this type of sticky mucus like thing accumulate within the lungs every time at every movement. The biochemical basis of cystic fibrosis is that, let us see, in normal persons, the chloride ions of the cell are pushed out through the participation of a protein called cystic fibrosis transmembrane regulator. So there is a protein called cystic fibrosis transmembrane regulator which pushes the chloride ion out from the cell. Now, those patients who are suffering from cystic fibrosis, this CFTR protein is non-functional. It is not produced due to its gene defect. So, CFTR must have been produced from certain gene, and CFTR gene function is what? To push out that chloride ions from the cell, right? It is a helper protein to move that chloride ions out from the cell. So those patient who is suffering from cystic fibrosis, the CFTR protein is not produced due to a gene defect that cannot able to produce this CFTR protein. Consequently, chloride ions concentrate within the cells of the lungs, which draws water from the surroundings. Okay. So what happened? The chloride ions concentrate within the cells, thus it draws water from the surroundings. As a result, the respiratory tract and lungs become fully dehydrated, and we have mucus all the time in order to remove away the dirt particles. But when that mucus become quite large, then we have a sneezing, coughing, that we know. But if that mucus is dehydrated, it becomes sticky, and which becomes an ideal environment within the lungs for having other opportunistic bacterial infections, which are the secondary infections, you can say. So simple thing, due to the simple thing due to the defect of a gene that is responsible for CFTR protein, it cannot able to move that chloride ions of the cell from the cells, and thus what happened? Chloride ions get concentrate within the cell. It draws water from the surroundings. Thus the respiratory tract becomes dehydrated with sticky mucus, which becomes an ideal environment for other opportunistic bacterial infections. Okay. Now, how gene therapy for cystic fibrosis is carried out? As the defective gene for cystic fibrosis was identified in 1989, researchers immediately started working on gene therapy for this disease because this disease is very dangerous. Okay, so the patients who is suffering from this disease, their gene has been identified that what is the gene responsible for causing this disease due to non-functional of their CFTR gene. Adenoviral vector system has been used, although the success has been limited. The major drawback is that benefits are short-lived since adenovirus do not integrate themselves into the host cells. So we can use adenoviral vector system by using their genome. The adenoviral genome has the non-essential part of the adenoviral genome is removed. In that place we use a therapeutic gene. Here the therapeutic gene is the CFTR gene, because that gene defect leads to cystic fibrosis, and then it is allowed that adenoviral vector to be properly integrated into the target cells. But the success has been limited in this case because adenovirus do not integrate themselves into the host cells. That's the problem. Thus, the patients are short lived. Multiple administration of recombinant adenovirus caused immunological response that destroys the cells. This is another problem. So as adenovirus is a outer vector, na, it is a virus, it is a antigenic part, it is a foreign body. So due to multiple administration of recombinant adenovirus, it cause immunological response which will destroy the cell. Thus, adenovirus is not employed in this way, and that is the main reason that why we use adeno associated virus, because we have seen little bit ago that adeno associated virus are non pathogenic, right? They are non pathogenic. It has been mentioned here. They are non pathogenic small DNA virus which can be used for treatment of cystic fibrosis. So it is saying here, by using adeno-associated virus vector system, some encouraging results were reported in the gene therapy of cystic fibrosis. In the phase 1 clinical trial, during the phase 1 clinical trial with cystic fibrosis patients, the vector persisted, that means it remained for about 70 days, and some improvement was observed in the patients. So, by using the adeno associated viral vector system, uh, we can we can introduce our target gene. Here the target therapeutic gene is CFTR gene into the patient, and it is employed by using two plasmids with an adeno associated virus. And it was in the phase 1 clinical trial, it has been observed that these such improvement observed among the patient because the virus remains stable for a long time. It remains stable for a long time, for about 70 days. So no immune response has occurred also, because it is non-pathogenic, right? Though it is non-pathogenic, it does not cause any immune response also. That's a good advantage for using them as gene therapy, and that's the main reason that why scientists are using adeno associated virus as a gene therapy. Now let us see about different another virus which can be used for in vivo gene therapy, which is your herpes simplex virus vector system. So the retrovirus and the adenovirus employed in vivo gene therapy are engineered to infect specific target cells. So they are engineered, they are modified, their gene is modified so that they can able to infect specific target cells. But there are also some other viruses which have a natural tendency to infect particular type of cells. So we can use such type of virus which can only be used for targeting that particular type of cells only. And the best example is your herpes simplex virus. Herpes simplex virus type 1 is infecting and persisting in non-dividing nerve cells. So it can infect and remain within the non-infecting target, non-dividing target cells, nerve cells. Herpes simplex virus is a human pathogen that cause cold sores and encephalitis. Okay, so normally herpes simplex virus, it is a human pathogenic virus which causes, though rarely it causes cold sores and encephalitis. Now, with the help of this herpes simplex virus, it can be targeted, it can be used as a vector to target in non-dividing cells, in non-dividing nerve cells. Now, there are large number of disease in a nervous system. Large number of human disease are found within the nervous system. So we can use this herpes simplex virus as a vector to target in the nerve cells for curing the disease. Like there are large number of disease like metabolic, neurodegenerative, immunological, tumors associated with the nervous system. So most of the some of the disease which are associated with the nervous system can be cured by using this here herpes simplex virus, herpes simplex virus as a vector. HSV or herpes simplex virus is considered to be an ideal vector for in vivo gene therapy of many nervous disorder. So nerve related disorder can be cured by using herpes simplex virus as a vector, because it is targeted, this virus is targeted to the non-dividing nerve cells. So most of the nerve related disorders can be cured by this herpes simplex virus by introducing therapeutic gene responsible for that cureness of this disease, and then this virus is targeted to the target nerve cells. Normally herpes simplex virus has a double stranded DNA of about 152 kb, so much huge as its genome. About 30 kb of herpes simplex virus genome can be replaced by a clone DNA without the loss of its basic characteristics. So, out of the 152 kb of herpes simplex virus genome, 30 kb of herpes simplex virus genome can be removed out, and in that place we can insert our therapeutic gene along with that clone DNA, along with that plasmid, without losing the characteristics of that HSV. Okay, but some technical difficulties in dealing with large size DNA in genetic engineering and experiments observed, because HSV is quite large, so there will be a little bit problematic when we are targeting that herpes simplex virus into a target nerve cell. Because of its large size, it is difficult to transfect them into the nervous cells. Although they are these are the targets, they are born to be target to the nerve cells only, but still some difficulty occurs. But otherwise, about 30 kb out of 152 kb of herpes simplex viral genome is can be replaced by our desirable gene, therapeutic gene, so it is beneficial. Herpes simplex virus vector could deliver therapeutic gene to the brain and other parts of the nervous system. So they are targeted to the brain only and to the nervous system only, related with the neurological disorder, related with the immunological tumors, metabolic disorders which are associated with the nervous system only. Okay, so these viral vectors can be used to employ um, to deliver its therapeutic gene to the brain and other parts of the nervous system. So as I have said before, these genes are well expressed and maintained for long period because they are targeted to that cells only, so these genes can properly integrate into the genome. There it will get expressed properly and maintained for a long time. More research is needed, however, before going for human trials. Still now animal trials is being going on, human trials has not been done. Up to that, we have used adeno associated virus system in human trials, where we have seen in phase 1 clinical trials, we have seen quite bit improvement to the patients who are suffering from cystic fibrosis by adeno associated viral vector system. Still, research is going on by using HSV as a vector in targeting to the nerve systems related with the disorders related with the nerve systems can be cured. But animal trials is going on. Human trials is not being done yet. Okay. In successful, if successful, HSB may be helped to treat many neurodegenerative syndromes like Parkinson's disease, which is common in India, and Alzheimer disease by gene therapy. So, if this herpes simplex virus can be successful in human trials, then we can use this herpes simplex virus vector for treatment of patients who is suffering from Parkinson's disease, who is suffering from Alzheimer disease, even multiple sclerosis, which is also a disorder in the nervous system, can be cured by using this HSV as a vector. Okay, till now, trials has been done in adeno-associated virus within the humans. Well, we can also go for non-viral vector systems also. We can use some artificial like lipid vesicles. Within the lipid vesicles, we can entrap our desirable gene, and we are targeting that lipid vesicles to the target cells where they will be engulfed, where they will be endocytosed, and it can deliver our desirable DNA. Okay, let us see. So there are some limitations by using this virus as a vector in gene therapy, because first is cost of production is very high, and most of the viral proteins can cause inflammatory response if immunogenic response occur within the host cells. So therefore there is a continuous research going on for using alternatives to viral vector system, and there are many ways through which we can deliver our therapeutic gene into the target cell. Like the first way is your lipoplexes. What is lipoplex? Lipoplex are the liposomes, you can say. What are liposomes? Liposomes are the small vesicles prepared from a suitable lipid. These lipids are used to form such lipoplexes, can be cationic, it can be anionic, it can be neutral, or a mixture of substances. Because of polyanionic nature of DNA, cationic lipids typically used for gene delivery. So DNA is polyanionic in nature. So cationic and neutral lipids are used for gene delivery. In order to form lipoplex, these cationic lipids often formed with neutral helper lipids, and most of the reagents used for making lipoplexes are cationic in nature. Okay, so lipid DNA complex are referred to as lipoplexes, or most commonly called liposomes. So we will have a liposome, lipid vesicles here. Within the lipid vesicles, we will have our desirable DNA. Okay, we will have our desirable DNA. Okay, now let us see what is saying more about the liposomes, lipoplexes. Now it is saying that they have a DNA construct surrounded by artificial lipid layers. So we have our desirable therapeutic gene, and that desirable therapeutic gene is surrounded by the artificial lipid bilayers, artificial lipid layers. A large number of lipoplexes has been prepared and being used. These are non-toxic and non-immunogenic. That means they will not cause any immune response when administered into the cells. Okay. So normally these liposomes are small vesicles which are prepared from suitable lipid, and because DNA is polyanionic in nature, cationic and neutral lipids are typically used for gene delivery, and to form lipoplexes, the cationic lipids formed with neutral helper lipids, we use reagent for this to make, like we can use dotma, DOTMA as a agent reagent for making lipoplexes. DOTAP, these are full form. Okay, then DOGS, the full form of DOGS is DIOC, dioctaddecyl. uh, I cannot able to recite the name here, it's not given in your book actually. So different type of reagents are being employed for making these lipids. Okay, like I am giving the name here. Okay, okay, I am zooming here to write the name here. Okay, for writing the DOTMA, the full form of DOTMA chemical name is very large, N 1 2 3 di dio sorry dialeoxy dialyl oxy propyl propyl, it has not been finished, N N trimethyl ammonium chloride. This is a cationic reagent used for making lipid transfection. Then another reagent is DOTAP. DOTAP full form is small 1 2 di dioleo leoyloxy dioleuloxy 3 trimethyl ammonium propane, trimethyl ammonium propane. Okay, another is dogs. Short form is dogs. Full form is, let us see, I trying to reciting this name, dio diotedicyl, this is C. Okay, this is C, CYL, amino amylo glycyl, amino, it's saying amylo glycyl spermine. So these are the some reagents which are employed for making liposomes, for making lipoplex. Okay. Now let us see more about the liposomes. So here we can see uh, how liposomes are being created. Next is DNA molecular conjugates. Okay. So how DNA molecular conjugates are being prepared, let us see. This is another approach. But by the way, I have not said here that some clinical trials with liposome CFTR gene complex shows that gene expression was short-lived. Now there is a drawback. We have used artificially created lipid vesicles by using some reagents like DOTMA, okay, then DOTAP, then DOGS, and we entrap that DNA, the therapeutic DNA, with the lipids, and we are administered these liposomes to the target cells. Okay, the target cells is, with the help of endocytosis, it the DNA gets entered, but it's getting to be degraded also. So some clinical trials has been done using liposome CFTR gene complex that shows that gene expression is not too much high. It's very short lived, not remain expressed for a long time. Okay, the next is DNA molecular conjugates. DNA molecular conjugates are what? Let us see. Use of DNA molecular conjugates avoid the lysosomal breakdown of DNA. Now, if we able to administer our desirable DNA into a target cell, so that the enzymes present within the cell, that is found within the lysosome, does not able to break down the DNA, to prevent them, we use this type of conjugates. Okay, that type of problem we have seen in liposomes, making liposomes. Another advantage of using conjugates is that large size therapeutic DNA can be delivered to the target tissue. So we can use large amount of our desirable DNA into the target tissue by using this type of complex greater than 10 kb. Okay, and the most commonly used synthetic conjugate is poly-L-lysine. So poly-L-lysine can be employed as a DNA molecular conjugate which is used for gene delivery. Well, poly-L-lysine is a positively charged synthetic polymer of the amino acid L-lysine. This poly-L-lysine facilitates the attachment of protein. It facilitates the attachment of cell by enhancing the electrostatic interaction between the negatively charged ions of the cell membrane. Okay, this poly-L-lysine here is bound to a specific cell receptor. Well, here you can see this poly-L-lysine is targeted to bound to a specific cell receptor. Then the therapeutic DNA is made to combine with, the therapeutic DNA is made to combine with them and form a complex. So here we can see it forms a complex, it forms a conjugate here, where you can see, if I mention this part, this inner part is the polyl lysine, where this is the cell surface receptor where it can interact with the target cells, and inside the purple color is your target DNA, the therapeutic DNA. Okay, now the therapeutic DNA is made to combine with conjugate them and form a complex. This DNA molecular conjugate binds to a specific cell receptor. Now it is allowed to bind to a specific receptor here, okay, to the target cells, and then it is engulfed by the cell. As you can see, the cell is going to be engulfed, the cell is going to be engulfed that conjugate, inside which now it forms an endosome. Okay, and thus the DNA from the endosome enter to the nucleus, where the therapeutic gene will be expressed properly without getting degraded inside the cell. Okay, so in simple words, if I say this poly-L lysine is used, it is bound to specific target cell receptor, and then the therapeutic DNA is made to combine with these with them and forms a conjugate and forms a complex like this. So it forms a conjugate like this, and then they are allowed to target to the they are allowed to transfect the target cells here. So here we can see, due to the presence of the receptor, it bound to the specific target cells where they are going to be entered by endocytosis, and they are not going to be degraded because of formation of this complex. They will be not degraded by the enzymes, so properly it will get be integrated in the target cells and into the nucleus. There it can be properly be expressed. Okay, now to enhance this whole process so that they can enter more rapidly, we for this transfection process we can make this process more faster by using some chemicals, chemicals like we can use polyethylene glycol to enhance this process. Polyethylene glycol is actually a polyether compound which destabilizes the plasma membrane actually, and it renders it permeable to the DNA. So here the DNA conjugate can able to enter faster by using the polyethylene glycol. It destabilizes the plasma membrane in the target cells here. Okay, and thus we can quickly allow the entry of this conjugate, this DNA molecular conjugate to enter. And inside the cell, this DNA molecular conjugate does not get degraded also because of having this type of complex. They cannot be degraded by the enzymes present in the cell. So thus the DNA released from the endosome enters to the nucleus, and after entering to the nucleus, the therapeutic genes can be expressed. Well, we can also use human artificial chromosome for in vivo gene therapy. I have already discussed with human artificial chromosome earlier that it is a small mini chromosome which is prepared artificially by assembling. Actually, it is just like an assemblage from other chromosomes. We are making a mini chromosome or micro chromosome. Okay, so it is saying here that human artificial chromosome or hack which can carry large DNA, one or more therapeutic genes can be carried along with some regulatory elements. It is a good and ideal vector. Studies concluded that cell culture using human artificial chromosomes are encouraging, but the major problem is that delivery of such large size of chromosome into the target cells. That is the main problem. In the ex vivo gene therapy, we have isolated the target cells, we have cultured the target cells, then we have transfect the target cell with human artificial chromosome. Here we will directly inject the human artificial chromosome into the target cells. That's also a big problem because of its large size. So nowadays researchers are working to produce cell containing genetically engineering human artificial chromosomes. Here, there exists a possibility of encapsulating and implanting these cells into a target tissue, but the way is long way to go. So we can encapsulate this human artificial chromosome by, like as I have said, by using lipoplexes or by using this type of conjugate where it can directly get integrated within the nucleus and get properly expressed. Okay. Now, what is the efficiency of using gene delivery by a non-viral vector? Well, viral vector are targeted or made to be targeted to infect the cells. But here we are using for all the cell types actually here. Here it is saying that the efficiency of transfection is quite low because we have to make specific interactions with the target cells, just like here for making the conjugate we use a particular cell receptors so that they can able to interact with the target cell, okay, ligand, and thus the efficiency is quite slow, which is not too much fast. Second is the expression of the therapeutic gene is very short lived. Consequently, there is no effective treatment of the disease. So, as the expression of the therapeutic gene, which are administered through the non-viral vector system, is not quite high, and thus it has to be done repeatedly, which is quite costly, and thus it limits the function. Okay, so for various diseases, we can either go for non-viral vector systems, we can go for viral vector systems, both in the case of ex vivo gene therapy as well as in vivo gene therapy. But specifically focus we focus on ex vivo gene therapy in case of viral vector systems, as well as in vivo gene therapy in viral vector systems only. There is a quite possibility, quite beneficial result has been observed. Like for instance, today we have discussed that how adeno associated virus is used for helping to cure the patients suffering from cystic fibrosis. Okay, now there is some other topics as well, which is given. Gene therapy strategies for cancer. Well, gene therapy can be used for treatment of cancer as well, and for other associated disease, right? But I will not discuss this one because there is some limitations here in your first semester syllabus. Okay, we are better to go for, you can study this, but it will not come in your exam in that way, because the only gene therapy has been given. It has been mentioned in your syllabus. If you see, so here you can see in your syllabus only your basic concepts of therapy has been given. I have discussed about the in vivo and ex vivo gene therapy in quite a long detail. That is enough for your syllabus. Well, most of the thing has been already discussed with you. Let us see if I get some time for some revisions or some important part which is left over. Okay, so that's all for today's lecture. If you have any doubts regarding the lecture today, you can ask me in the WhatsApp group for any query related with the lecture today that I have provided with you.